Hi, everybody. I'm Charmei Tang, President and CEO of Creative Microtech. I'll be presenting to you our revolutionary LifeTrac assay applicable to broad spectrum of cancer testing. LifeTrac assay transforms cancer diagnostics, saving and improving lives. The LifeTrac assay platform has very broad applications because the biomarker is applicable to all solid tumors, all stages of cancer, and all therapies. The assay is applicable to cancer screening, companion diagnostics, monitor therapy, predict treatment response after 1 or 2 treatment cycles, predict aggressiveness of the cancer, detect residual disease, and early detection of cancer recurrence. Our lead application will be lung cancer because patients with lung cancer needs help. In the US, the number of new cases per year is about 240,000, and the number of deaths per year is about 154,000. This is 25% of all cancer deaths. There are a number of unmet needs LifeTrac tests can fulfill. We can replace expensive and high-risk lung biopsy to confirm cancer and for companion diagnostics. LifeTrac tests can determine disease progression earlier than imaging. Immunotherapy advanced lung cancer therapy significantly because approximately 25% of lung cancer patients achieve long-term survival, but still not for everyone. The cost of immunotherapy, however, is very high, on the order of $150,000 to $300,000 a year. LifeTrac assay provides a solution that can benefit the patients by providing safer and cheaper blood tests. The clinical benefit of LifeTrac tests can provide also more accurate complementary or companion diagnostics to determine potential effectiveness of immunotherapy by a blood test. The ability to identify non-responders to immunotherapy in only 30 to 60 days, much earlier than imaging, it allows the physician to proceed faster to step therapy. Our LifeTrac tests also provide savings to patients and the insurance companies. We identified a type of cell in the blood of cancer patients. We named the cancer-associated macrophage-like cells. We call it CAMOs. They are macrophages that engulfed tumor cells and tumor debris in the tumor and returned to the bloodstream. They are polynucleated and they are very large, 25 to 300 microns in size. And as you can see here, have a cell that's white blood cell with arrow pointing to it, and they are much smaller than the CAMOs. And the CAMOs have funny shapes. They, when they are small, they are round, and they can grow one tail, or they can be 2 tails. And they are very large, and you do not see this kind of cell in healthy controls. We find in also solid tumors, we find them in all stages of the disease. And they are, because they engulf tumor cells, they have the markers of tumor proteins, tumor DNA, and tumor RNA. And they are M2 phenotype macrophages. We collect them by using our cell-sieve microfilters. This slide presents data from 293 patients from 6 different cancers on cancer CAMO biomarker, and we compare the data to circulating tumor cells. CAMOs are found in all solid tumors and even as high percentage in early stages, but not CTCs. We are the only company with a device that can consistently capture camels, and we are the only company obtained clinical application of camels. One of our discovery is that camel size provide a significant clinical information. This slide shows camel size can provide prognosis if the patient has a camel greater than 50 micron in size. The patient has significantly shorter progression-free survival and overall survival. Camel size can also predict treatment response within 30 to 60 days of start of therapy. The data presented here is from a stage 2 and 3 non-small cell lung cancer patients treated with immunotherapy with chemoradiation. The progression-free survival for patients with camel size bigger than 50 microns is 8 months, while patients with camel size less than 50 micron is 24 months. This is a significant difference, and the p-value for the progression-free survival is 0.0016. We have significant amount of scientific data to support our claims. We have found camel in more than 20 different solid tumors. We are conducting 15 clinical trials. We have received 4 research grants from NIH and Department of Defense, 3 of them on cancer screening, and 1 on predicting treatment response for lung cancer. We have 15 journal publications and many in progress. We have filed 11 PCT patents, and we have full ownership of all the IP. The leadership team at Creative consists of myself and Peter Armstead. I'm the founder, president, and CEO. I got all my degrees from MIT. Pete Armstead is the CFO and VP of Business Development. He also got all his degrees from MIT. We have an excellent scientific advisory board. Massimo Christopher Finelli from Northwestern University. He's a breast cancer oncologist. He was on our first camel publications. We collaborated with Stephen Lin from MD Anderson on lung and esophageal cancers on many different projects. Christian Hausenchild, he's a pathologist. He advised us from the very beginning to accurately identify each type of cell that we capture on our microfilter. He helped us to distinguish camels from other cell types. Our commercialization plan consists of 3 parts. The first is to set up a clear lab, and this allows us to generate revenue. Next, we want to develop automation. The purpose is to enable processing high volumes of samples and to reduce costs. There are 2 components. First, automation for blood sample processing. Secondly, reading slides utilizing artificial intelligence. The third part is to start pivotal FDA trial. The goal of the trial is to predict treatment response within 30 to 60 days after start of immunotherapy for unresectable stage 3 non-small cell lung cancer, and also to obtain data from the same patients for concurrent CMS review to get reimbursement. FDA approval will enable our diagnostic to obtain wide adoption quickly. We want to raise at least $20 million. The funding will start the FDA approval process, but not necessarily sufficient to complete the process. We aim to obtain FDA approval in about 3 years from the start of FDA process. At the start, we will set up a CLIA lab and start automation. Our lead FDA-approved product will be for lung cancer. The addressable global market for lung cancer is more than $1 billion. The LifeTrac test addresses many unmet medical needs. There is a minimal competition in the blood diagnostic market to predict immunotherapy treatment response. We have a broad spectrum of patent protections for this diagnostic. Our long-term vision of our LifeTrac test is very bright. The global market potential is very large. LifeTrac test is applicable for cancer screening, cancer diagnostics, and cancer recurrence. Initially, our CLIA lab will focus on cancer recurrence testing. The market size is total greater than $35 billion. We will apply for FDA approval for diagnostic tests, starting with immunotherapy for lung cancer, and we will continue to apply for additional FDA approvals for other diagnostics. Finally, cancer screening has the largest market potential, but the cost of FDA clinical trial for cancer screening will be very expensive because it requires very large number of cohorts. And we will be doing this in the future. Here is a partial list of our research collaborators and some clinical trial partners. Finally, here are our contact information. I'm showing also a strange-looking camel with 2 tails. It's unlike any other cells you find in the body of a patient. Please note that our website is creativebio.com. Thank you very much.